first heard about Dr. Pietsch from Frank Corsetti from the art department, who said, you've got to get in touch with her. She's at San Jose, great researcher. So I'm very excited you could come up. Came to LA originally roundabout through New York, growing up near the city, came out undergrad at Cornell, which is an amazing paleo place actually. It's gorgeous, as they say. And you definitely didn't know that. And it was USC for a PhD with David Botcher, who's just a giant in California paleontology. Back to Cornell for a postdoc at the Paleontological Research Institute, and has been at San Jose for 3 years. And I thought she'd worked on KT boundary stuff, but not really. But extinctions are her passion, and what we'll hear about is some work she's done on the Paleocene-Eocene Thermal Maximum. I did it?
Perfect. Amazing. Yeah.
Thanks for coming.
I was going to say, I'm like, Jason is a literal giant, a very tall man also. So if you've ever seen Big Butcher in real life. Yeah, so this is work that I did as part of my postdoc and sort of wrapped up when I started at San Jose State. So I'm going to talk to you today about the Paleocene-Eocene Thermal Maximum, and if you haven't heard about it before, I'm going to set the stage for you. And the title is going to give away the end conclusion, right? Shows little impact on shallow marine mollusks. So get ready for 45 to 50 minutes of how to spin your work when you find no change in the fossil record in response to an extreme temperature event. What an exciting find. So the first slide I always put up is this work is, I was just a part of. It's based on a grant that Warren Allman, who was my undergrad and my postdoc advisor, wrote with Linda Ivney, who's at Syracuse, and Rowan Lockwood, who's at William & Mary, to look at the Paleocene-Eocene Thermal Maximum and other deposits in the Gulf Coastal Plain of the United States. And Jocelyn Sesso was a postdoc with them before I came and worked on this project. And John Handley is now faculty at Rochester University in upstate New York, but at the time when I was working with him, he actually worked for Xerox Corporation in logistics, but he has a soft spot for paleontology and is a bastion of data analytics, loves Bayesian statistics, and just loves to find problems to apply his love of paleontology and statistical knowledge to. So how lucky are we? So much of the statistics that I'm going to talk about today I learned from John, and I'm always inspired to learn more. So now he has a faculty position so he can talk to more students about this kind of work. So I wanted to start out introducing the early Eocene, which is the time and place that we're going to be talking about today. So this is our modern continental configuration, and in a second I'm going to show you an early Eocene Earth. And I want you to make a comparison of what these two globes look like, and tell me some of the differences that you see between the Earth today and the early Eocene Earth.
Can you flip back and forth a
yes,
couple times?
You can do a couple back and forth. So here's the Earth today, here's the early Eocene today, and the Eocene. So just big picture things, right? Like, don't worry about what Florida's doing necessarily.
The Atlantic Ocean is smaller.
Yes, definitely. The Atlantic Ocean has expanded substantially since the early Eocene. What else do you notice?
It looks warmer, like I'm seeing glacial stuff.
Yeah, no ice at the poles. Definitely a warmer time. Any other differences you notice?
Yeah.
Yeah, here's India, a beautiful island out in the middle of the Indian Ocean, or the Proto-Indian Ocean. Yeah.
North Atlantic's all blocked up.
Yeah, that's very true. Really small up there, and then I did a bunch of work at the Paleocene boundary for my undergrad, and so I always am looking for, what is the connection between North and South America, or lack thereof. Yeah, but what I definitely wanted was the ice comment, which usually doesn't come so early, so well done. I usually have to drag that one out. So it's really warm, the PETM, Paleocene-Eocene Thermal Temperature Maximum, is a really hot time, and that means the Eocene was a really warm time. So you're used to the tropics being at the equator, a very warm area, these arid regions that we have today at 30 degrees north and south. But what's different about the early Eocene Earth is what's happening at the poles. So places today that would be temperate, like upstate New York, would become paratropical, and then the North Pole was warm temperate. So when you've heard stories about people finding crocodilomorphs and palms at the poles, this is the Eocene Earth. So what's happening here is a differential warming. Most of the warming is not happening at the tropics. You only have a couple of degrees C increase in temperature here at both the land surface and the ocean surface, but a lot of the warming is happening at the poles. And you may have even seen in some of your classes how we have outsized warming at the poles in response to modern anthropogenic climate change. So we have a decrease in the temperature gradient. As you move today from the equator to the poles, it gets substantially colder, but the poles will warm to sort of even out, have like a flat line for a temperature gradient or less of a curve. So that's what we're seeing at the PETM. We can look at this through time. So on the y-axis here, we have 70 million years ago. Here's the KPG boundary, and then the rest of the Cenozoic. And the PETM really stands out if we're looking at oxygen isotopes, which we're using as a proxy for temperature, as this really rapid spike in global temperatures. Okay, so it's really fast, and it represents one of the warmest intervals in the last 66 million years. So it's an extreme temperature event that happens really rapidly. So this is work by Zakos in 2001, who's continued to work on this boundary. So what does this evidence from the geochemistry look like? When we're looking at the oxygen isotopes, which again is our proxy for temperature, you can see this really rapid negative isotope excursion. So that's indicative of the warming. But they want to know something about the mechanism, right? Like what is causing this extreme warming event. So they're looking at carbon isotopes to get an idea of what is the source of this warming. Could it be addition of carbon into the atmosphere? Probably it is. And so you see this negative isotopic excursion in carbon isotopes, which could be a lot of different sources, right, of carbon isotopes. And so one of the hypotheses they have for the PETM is the loss of methane clathrate deposits. So these clathrates are sources of methane that are frozen, that if they get disturbed, will sublimate up into the atmosphere, put a lot of carbon into the atmosphere and cause warming. But what is causing the disruption of those clathrates, that's still sort of up for debate. Is that volcanic events that are warming the ocean, sort of reaching a tipping point? Was there, I've heard hypotheses of an asteroid impact coming in, right, and disturbing these clathrates. But the isotope signatures can be used to model both the rate and the amount of carbon at what isotopic signature would have to go into the atmosphere to sort of figure that out. And that's a lot what Zakos has done. The other thing that's really interesting is a change in the calcium carbonate content. So this is a marine core in particular, but you can see these changes in the oxygen carbon isotope space in terrestrial systems as well. But in the marine setting, we see a loss of calcium carbonate content. So that's indicative of a change of the ocean's pH and of the carbonate saturation. And so if you are a shelly mollusk, it's going to make it hard for you to potentially build your skeleton during this time. So that's all the geochemical evidence for change, yeah.
One aspect, if it's warmer, wouldn't it, not the pH, but the temperature should actually enhance carbonate productivity?
Could, yeah, yeah. But there's pretty good evidence for acidification in some situations here, but I want to talk a little bit about evidence for buffering also at the end as well. Okay, so I like to talk about the rates of carbon addition. So this is a more recent paper by Penman and Zakos, again, trying to do this modeling based on the isotope signature of the carbon, isotope excursions, and playing around with different sources of carbon that they're putting into the atmosphere to try to figure out, sort of backtrack what it could be that would have been changing the atmosphere and the oceans at that time. And so what they're showing here is the modern rate of anthropogenic carbon input into the atmosphere and the PETM rate. So you can see in the modern, we put about 10 gigatons of carbon into the atmosphere every year. That's this red line. And the PETM is this black line, which you can almost barely see here, right? At only 0.1 gigatons of carbon per year. So a much lower rate of carbon input. And what that looks like if you're going to model atmospheric PCO2 concentrations is what we're seeing right now, this really rapid rise in PCO2 and then it leveling off, compared to the PETM where that PCO2 concentration rises steadily from this lower rate of carbon being put into the atmosphere. And so we like to look at the PETM as a model for what might be happening in our modern climate change to suggest if we see certain responses of plant communities, terrestrial vertebrates, marine invertebrates, like I'm going to talk about today, what might we expect? But when we look at the PETM compared to the modern, it's not really an analog, right? It's the closest thing we have in the geologic record in terms of the rate at which carbon was put into the atmosphere. It's quite fast when you look at all the other isotope signals of the last 66 million years. but it's still about 10 times slower than what we're doing right now. Yeah?
What's the x-axis on the bottom graph?
Oh, I think it's, ooh, why do I have a box there? Why did I do that? This is, by the way, thousands of years.
Oh, okay.
I'm gonna guess that it's the atmospheric, it's the atmospheric PCO2. I think,
Oh, no, you answered my question.
Yeah. Okay, yeah.
All right, thank you.
but I'm wondering like, why is this here and not here? And I'm wondering if the red line and the black line have different axes now. And I'm wondering if I just like covered over it. Because I see this 8.2, which to me is pH, right? So I'm wondering if there's a pH thing that I covered when I made this originally. Anyway, I don't want to like have too many lines going across the screen either. Okay, so what you're all here for is to understand a little bit about the biology of what changes in response to the PTM. So there's been a lot of research into this boundary since that isotope excursion was discovered, and what we see for a lot of the plankton fauna is migration. So dinoflagellates, planktonic forams moving towards the poles, so trying to move with the increase in temperatures, get away from the superheated tropical environments. And extinctions of coral reefs. So this orange area indicated here, we see extinction of corals and replacement of those corals by nummulites, which are these large coin-sized benthic forams. So they're building reefs during the Eocene instead of corals. When we look at the terrestrial environment, this is just a model that represents changes that have been observed in the Bighorn Basin in modern-day Wyoming. And what they see is before the carbon isotope excursion, a forest dominated by coniferous plants, and during the body of the carbon isotope excursion, so that's excursion is representing about 100,000 to 200,000 years, they see a change to these more open, drier savanna grasslands that are adapted to the increased seasonality that the higher temperatures potentially bring, as well as the increase in temperatures. But then, as the carbon isotopes, whatever the disturbance is, we see a shift back to the sort of normal background carbon isotope signal and a return to these coniferous forests again. And what's really exciting about this is they see this basically as evidence for migration, right? Of the pine forests are moving north, these grassy shrublands are moving up, and then everything shifts back down. And there's only about 5 groups that go extinct in response to this, and about 5 groups that migrate in and stay, even when the pines come back. But they're not seeing a massive extinction at this time, just this slow and steady response, this shift of the biomes. When we look at terrestrial systems, looking at mammals, this is a really interesting time for mammals. All of these bars here represent a different genus of mammals, again in the Bighorn Basin. And so the gray bars here represent mammals that go right through the PETM, no change. The red are groups that are going extinct, but you'll see that they're not really necessarily lining up at that boundary exactly, but you know about signal ellipse effects and everything like that. But what's really cool are all these green lines, all these new species that appear in the earliest Eocene. But these aren't originations. These aren't new species evolving. These are species that are migrating, because remember how warm it is? Now you have these land bridges. Groups are coming over from Asia, so we get the arrival of primates, artiodactyls, and perissodactyls into North America during this time. So all of our hoofed mammals and primates start to appear in the early Eocene. That's what's happening there. So there's lots of different changes that are happening to all these different groups: plants, mammals, corals, plankton. So what's happening to marine invertebrates that live on the shallow shelf, which is what I study, it's what I'm interested in. And so this review paper that was all about the PETM in 2012 said the response of most marine invertebrates, mollusks, echinoderms, brachiopods, to paleoclimate and associated environmental changes like acidification or deoxygenation during the PETM is virtually unknown. So read your review papers and look for things like this, because you could do entire projects based on what people say is virtually unknown. And the reason for this is that it's really hard actually to find a shallow marine section that preserves the boundary. So that's where we're going to go. This is what Linda and Rowan and Warren were all looking at, was the Gulf Coastal Plain of the United States, and there are also Atlantic Coastal Plain sections that preserve Paleocene and Eocene deposits, but we're going to focus on the Gulf Coast. And specifically, a couple of units, fossiliferous marine units, in Mississippi and Alabama, because this allowed us to have really good control of the stratigraphy and understanding of the timing of the different depositional events. There are also Paleocene and Eocene units in Texas, but it's a little bit harder to correlate them exactly to the Mississippi and Alabama sections, so we didn't really focus on those. So the geologic setting during this time, the Gulf Coastal Plain, if you know anything about the geology there today, is very similar in the past. It's an area where you have a lot of transgression and regression, so sea level rise and fall throughout these intervals, and a proto-Mississippi deltaic system. So the delta is migrating around. So you have these deltaic and interdeltaic deposits during this time. And I did a whole paper about glauconite and interdeltaic deposits and fossils that I will not get into. It's a little drier. It doesn't make for a thrilling seminar talk. So these are the units that we're working with here. We have 3 units from the Paleocene, the Australia-Thiersay beds, the Greg's Landing Marl, the Bell's Landing Marl, all from the Paleocene side, and then the Bashai Marl and the Upper Hatchatigbe from the Eocene side. And each of these fossiliferous units is about 0.2 to 1 million years in duration. And what you'll notice is that we don't have one spanning the boundary, right? We have about 1 million years between this last Bell's Landing Marl and the Bashai Marl. The boundary is here. There's a carbon isotope excursion, but it's in terrestrial sediments. So you can see that boundary. You can actually see the primates that begin to fill in the terrestrial biome, all here in Mississippi and Alabama, but we don't have the marine units there. So we said we still want to look at this section because there is, at this point in time, no shallow marine mollusk fauna that actually appears at the boundary. Nobody has found that geologic section yet. So this is sort of the best case scenario that we have, is this 1 million year time gap. But if you are looking at the other groups of animals we've talked about, right, the plankton, the plants, the mammals, we do see massive and lasting changes in some of those examples, right? Major migrations, major extinctions. So even if we don't preserve the boundary itself, we might expect to see some big change on the Eocene side of things. So that's what we're going to go out and look for. But bear this in mind as we go through the rest of the talk. So we have 3 approaches. One of those is to look at diversity. Do we see a change in the makeup of the species that are in the Paleocene versus the Eocene? The other is to look at abundance, because if you think about what are the most abundant groups in a community, those are going to be the ones that control the ecology. And then the other is to look at life history changes, things like growth rate, body size, to see if those respond to this extreme warming event. So the first thing we're doing is this diversity structure. And so Jocelyn Sessa, who was the postdoc on this project initially, and Linda spent a lot of time going through the literature, entering data into the paleobiology database, going through collections, entering that data into the paleobiology database. And when I came on, the only thing that really was lacking was that last Upper Hatchatigbe unit. So this last unit here in the Eocene. So I went down to Mississippi and worked with David Dockery, who's at the Mississippi Department of Environmental Quality, which is where they keep all their fossils from Mississippi, and looked at and identified some additional Upper Hatchatigbe units. So that's the photo that I sent around. That's me in the shack that they keep all these fossils in. It was a little disheartening. I was like, oh, hope we don't have a really big storm. Speaking of tornadoes, that was a fun time to be in Mississippi. I was like, oh, okay, very small building. So this is where John comes in, is looking at the various ways we can analyze the diversity structure here. So we're going to look at raw diversity, we're going to look at rarefied diversity, and we're also going to look at sample coverage. So you can see our 5 units here, 3 from the Paleocene, 2 from the Eocene side, the duration of these different time bins, and what's really important is the number of collections within each of these. So you'll see that one of these things is not like the other. One of these units is a little bit longer than the others, but not by too much, not too much compared to the Gregs and the Bells, but really over-collected. The Bashai is pretty fossiliferous. It's got lots of really interesting taxa, and so paleontologists have been over-collecting it for a really long time because they just are so curious about it and they want to get everything out of there. Versus the Greg's Landing Marl, actually, at this point in time, there's no known locality where you can go and collect the Gregs at this point. It's been filled in with river dams and construction sites. So you can't go out and get more information out of the Gregs. You're dependent on what's already been collected. So because of these differences in the sort of paleontological interest and the physicality of these different beds, we're going to rarefy our samples because we can compare this raw diversity, but I can bet you that the Bashai is going to be much higher than these other units just based on the effort that's been put into those beds. So rarefaction just allows you to find a place, a number of individuals that you can say is relatively similar between these different samples that have experienced different levels of collection. So that's one way we're going to compare these samples, is a rarefied diversity. The other is this idea of sample coverage. So this is figuring out, if you were to be collecting at the locality, or maybe you're collecting from a data set that you already have, right, just kind of pulling out of your bag of samples, what's the probability you're going to find a new species if you add one additional individual? So this is looking at, and there's a lot of curves and colors here, we have our 5 units from the Paleocene to over here from the Eocene, and we're looking at the entire data set, and then we're looking at the 4 largest samples from each time bin. And so you can see that, of course, the the Bashai marl, which is the orange line here, is the one that's been over-collected through time. So it has great sample coverage. But when you compare sort of like the 4 largest samples from each time bin, you get the sense that they are similarly diverse. So if you sort of just like take the Bashai down a notch, it's comparable to these other units. And that's the plot that I'm going to show you here. In the gray bars in the background, we have raw species richness, and as predicted, the Bashai's species richness is extremely high. But when you look at rarefied or the sample coverage across the boundary, you see that there's not a big change in diversity or richness, taxonomic richness, from the Paleocene into the Eocene. We don't see a big change, and if anything, we see an increase in diversity moving into the Eocene. So first things first, like we're experiencing this extreme thermal maximum, we might expect to see extinction here, and that's not what we're seeing in terms of just the raw diversity. But you could have diversity remain really steady, but have a lot happening underneath the surface. I like to equate this to a tsunami moving through the ocean. When it's out in the open ocean, it's not a big wave, it's just this rush of energy. So you can have all sorts of extinctions and originations sort of happening, but the line will look steady. So then we're going to look at is to see how many extinctions occurred and how many originations occurred. We'd expect to have a high extinction from the Paleocene into the Eocene, so the greatest extinctions in these Paleocene units, and the highest origination or new species in the Eocene units. And do we find that? So that's our diversity curve. And what we see here in terms of our first appearances, so this is origination. We actually see more origination in the latest Paleocene, in the Bell's Landing marl, and lower origination in the Bashai marl. So that's not what we expected. If anything, we'd expect the Bashai to be high. And in terms of extinction, we expected a high extinction value here of last appearances in the latest Paleocene unit, the Bell's Landing, but we actually see that in the Gregs. So we're not seeing major turnover from the Paleocene into the Eocene in terms of taxonomy. And these values of last appearances and first appearances aren't really outsized compared to the rest of the time bands that are readily available in the Gulf coastal plain. You can take the stratigraphy from the Cretaceous into the later Eocene, and these rates of change are not really different from any of the other boundaries, both mass extinction-wise, but also just sort of like normal turnover. So actually we see the greatest turnover within the Paleocene, with our 2 biggest changes between the Gregs and the Bells, and not at the boundary itself. Okay, we also looked at the taxonomic composition just to get an idea of what are these taxa, because again, we're working with data that's been put into the paleobiology database, so it's really easy to just like download this Excel file and like run these statistics and then be like, nothing changed, but you want to kind of maybe know what your critters are, which is sort of moving us into the ecology side of things. Okay, so like nothing is changing taxonomically, the diversity is the same, extinction origination isn't changing, but it's something about the abundance changing between these groups. So we have no change in taxonomic richness or turnover of benthic mollusks. We have this big hiatus though, right? We have this 1 million year interval. So there could be lots of interesting things that change in that million years that we just don't preserve, including we could have a major migration, right? Everything could have sort of shifted north and come back down again in 1 million years. The plants are doing it in a couple 100,000 of years, and we're just not picking that up. So there's still questions, right, because of that, that gap. But we're sort of banking on this, we're expecting a lasting change in some way, shape, or form, either in diversity, ecology, or life history. So then we're going to look at the ecological guilds, and this was my part of the project for sure, because that's what I like to look at, is the ecology of benthic mollusks. So we're going to code each of these groups for what they do, how do they move, what do they eat, how do they interact with the substrate, and look at abundance, because the things that are the most abundant tend to have an outsized effect on the ecological structure. And analyze these with ANISIM, which is analysis of similarities, and a mixed effect model. So there's a couple ways to make a living as a benthic mollusk, and maybe you already know some of the things. You can take in paleo. You can be infaunal, which means living in the sediment, epifaunal, living on the sediment surface. Semi-infaunal is sort of in between, right? Or you could be attached with byssal threads, like a modern mussel, or making a reef, like an oyster. You could be stationary or actively mobile, like this carnivorous gastropod over here. And in terms of the diet, we have herbivores, we have carnivores, algal scrapers, some chemosymbionts, all making up the different benthic guilds. So this analysis of similarity is really great for comparing these ecological groups. It's looking at ranks within the group, so how the abundances change within the group, all the different samples pulled into these time bins, and then how do they compare to one another. And particularly we're using this Bray-Curtis distance matrix, and that's pretty common for NSM and lots of other sort of plotting of multiple, like if you do like non-metric multidimensional analysis, it often uses the same kind of distance measurement. So this is the result that you get out of the NSM. 3 units here are from the Paleocene side, 2 units here from the Eocene side, and if you're looking at this, you could just sort of say like, I'm not seeing a lot of change here, right? It's not a big trend. The dissimilarity within each group is very similar to those across, and this low R value tells you there's very little difference between these samples. So as you're moving from the Paleocene into the Eocene, we're not seeing a big change in the overall ecology. And this is everything kind of binned together, but I'm really persistent. So I was like, let's look at individual classes, like just their diet or just how they move through the sediment to see if anything changes. In order to do that, we wanted to have some statistics to say, is there any difference between the Paleocene side and the Eocene side? And so John developed this mix effect model. So this is looking at the proportion of infinality, so things that live in the sediment compared to things that don't. So there's this proportion on the side here. And what you're seeing are the gray bars represent the averages for each of these different time bins we've been looking at. The 3 from the Paleocene and 2 from the Eocene side. And each of these lines represents one of the collections that was made. And these collections are kind of tied to one another because even though they might be independent units, they're collected maybe by similar people, and they're definitely collected from similar localities, right? It could be people over the decades, but they're all going back to the same riverbank. So these collections aren't necessarily independent from one another. And so the mixed effect model takes that into consideration in order to assign you confidence intervals and p-values when you're comparing things that sort of have this connection between them. So that's what I'm going to show you, and I show you all of these charts. Those error bars come from these mixed effect models. Okay, so this is just looking at motility. Does anything change across the boundary here? And you'll see that most of the groups are facultatively mobile before, and most of them are facultatively mobile afterwards. The only thing that really changes is the Australopithecus beds are mostly made out of oysters, stationary attached, and so when you lose those oysters, you see that big change, but not across the boundary itself. When we look at life habit, we do see a significant change. So we see a change in the proportion of infaunal groups and a decrease of epifaunal groups across the boundary. So from the Australopithecus beds into the bells, across the boundary into the Bashae, these are the epifaunal groups decreasing, and these are the infaunal groups which are increasing. In addition, the semi-infaunal and shallow infaunal are taking up a larger proportion as well as you move across the boundary there. So we do see a significant increase in organisms living within the sediment as opposed to on the sediment surface. And then the last thing to look at was their diet. Most of these groups are suspension feeders, you can see across the boundary. We have a decent amount of carnivorous gastropods. And the thing that appears on the other side are these chemosymbiotic bivalves, which when I saw this result, I was like, oh, how exciting. Something's different on the other side of the boundary. Wow, after all this no change. So these are the cyanid bivalves that are thought to be chemosymbionts, and so you might be thinking like, oh, maybe it's a nutrient-starved time, and so these chemosymbionts are sort of taking advantage of this opportunity they have to get really abundant in this place where they're a little bit more well adapted. So how do we interpret all these changes, or lack thereof? We do see this increase in infaunalization, which was interesting. And you might think it could be a response to lower oxygen concentration. So when temperatures go up, there's less dissolved gases, oxygen concentrations are going to be slightly lower. And so infaunal groups that live in the sediment might be adapted to those conditions in the first place, and so might be able to thrive, whereas epifaunal groups might not. So that's one possible explanation, but the other is that there is a trend of infaunalization from the Cretaceous into the Cenozoic. And so distinguishing whether or not this is a response directly to the PETM or just part of this larger trend is really hard to say, since when you look at it, as kind of stepping through all of those beds. When we saw those chemosymbiotic bivalves, I was really excited, and then I read some papers by Koznick, who'd done all of his PhD work in the Cretaceous and the Eocene, and he sees these chemosymbiotic bivalves pop up now and again. So they don't seem to be related to the PETM itself, and they don't seem to be related to particular conditions. They just get abundant now and again. Still have questions about that. So if anyone ever wants to work on chemosymbiotic bivalves in the Gulf Coast, I'm curious. So overall, we interpret this to say that the PETM in the Gulf Coast is not showing selection that favors any particular physiology. We can't really hang our hat on any of these trends, meaning something about the PETM itself. But it seems like the groups are pretty active in the first place. They're facultatively mobile, they're suspension feeders, and they're doing well on both sides. So the last thing we want to go for is this life history trait. Does anything change about the way they grow, about their body size? So I came from the Permian-Triassic boundary, and in the Triassic side, the recovery of the PT, there's this huge body size decrease. So I was like, maybe they get smaller. And there's been research done on the mammals that show the mammals are quite small in the earliest Eocene. There's dwarfing in some of the plankton, and so I was like, maybe things got tiny in response to this. Okay, so we're going to look at two groups. One of those is terrestrial gastropods, and the other is venericard bivalves. And we're choosing these because if I go back to that slide with all the colors and the names of species, Turritella and venericards are some of the most abundant groups in the Paleocene and the Eocene deposits. And also Warren Allman, one of the PIs on this, is an expert in turritellas, and Rowan Lockwood is an expert in venericard bivalves. So there's a little bit of bias, favorite study organisms, but they do make out an outsized proportion of the fossils that you find here. So we're going to look at size frequency distributions. What is their body size before and after the PETM, and then do stable isotope analyses to see how does their growth change or not across the boundary. So finally, a positive result. Turritellas get smaller across this boundary. So here is length, and here are deposits from the Paleocene side and the Eocene side, and you'll see that mean and maximum size of turritellas does sort of decrease across the boundary here. Not great data. I have some better data to show you on the next slide. But when we look at venericard bivalves, mean and maximum size, no change, right? And this is interesting because even though one is a clam and one is a snail, they do really similar things. They're both semi and faunal suspension feeders, so we were sort of like, oh, maybe they'll respond similarly since their ecology is really the same, but they are not. Okay, so why are turritellids smaller on the Eocene side? Well, we don't see a lot of turnover, right? We don't see a lot of extinction or origination at the Paleocene-Eocene boundary. We do actually see that in turritellids. So statistically, as a whole group, as a whole community, we don't see a lot of change. We do see extinction of turritellids. The large groups here do go extinct before the boundary, and then the groups that originate on the other side of the boundary tend to originate at smaller body sizes. So here are our Eocene taxa versus our Paleocene taxa. This is the shell length. So, 48 millimeters here, so about 50 millimeters, about 5 centimeters, down to about 2 centimeters on the other side of the boundary. That's data that came out of the collections at PRI. So let's look at the growth rates. So if someone gets smaller on the other side of the boundary, how does it do that? One way you could get smaller would be to, what could you change about yourself?
That's a snail, so the actual body diameter can change.
Yeah.
The juicy parts.
The juicy part, which is hard, right? Because we're just doing the shells. Yeah, so much could happen with the juicy parts. You never know. Right?
Well, but that'd be reflected in its body.
It'd be reflected in the shell.
Right.
Right. How do you get big? Like, what are the ways to get big?
More oxygen?
Oh, okay. I'm thinking like in terms of like rates. Like if you want to have a really big body, what are your options?
Steroids?
That would work for a human maybe, or a mammal. For a snail, you could grow faster, which I guess is kind of like maybe steroids a little bit tangentially related to. So you could grow faster, or in the case of invertebrates, which we don't think about how invertebrates live all the time, you could live a longer time because they're sort of doing this indeterminate growth thing. So they live longer, they'll just keep getting bigger potentially. Some of them, not all of them. And turritellids tend to grow to a sort of an adult size and then slow down their growth after that. So we're going to look at their growth rates, right, in the Paleocene and then in the Eocene. So this is the Paleocene side of things, and Mary Kozlowski was a graduate student of Warren's who started collecting this data set. And so these are two different individuals, the black line and the gray line here, and this is their oxygen isotope values. And you can see that these individuals live about 1 going on 2 years, right? So they're going through these summer-winter cycles reflected by the oxygen isotopes. And they grow to about, so this is their spiral distance, so this is measuring like around the spiral, because that's how you're taking the isotopes to reflect their ontogeny, from their larval stage all the way down to their adult size. They're getting to be about 350 millimeters in that spiral distance. Okay, so then we're going to compare this to the Eocene samples. And on the Eocene side of things, what do you notice is different? How long do they live? How old are these? If these are 1 going on 2 years, how many sort of cycles?
1 year.
Yeah, about a year, year and a half, right? So they're living a little shorter, and then how fast do they grow? Like in a year, this one gets to be about 200 millimeters. And in a year, this one only grows maybe 100 millimeters, right? So it's a lot slower in the Eocene. So they're growing a lot slower, and they're not living quite as long. So that's sort of the mechanism for how you be small on that side of things. Okay, so that data had already been collected before I started doing my postdoc work, and I was like, well, what about the clams? So then I went and worked with Linda Ivney and collected all this data for Venericord bivalves. And so I did this with her graduate student, Emily Judd, who knows how to use this micromill and taught me everything I knew about isotope sampling. And so this is really time consuming. All of these lines on here are these individual isotope samples. So I was joking earlier, well, it wasn't a joke, I watched an entire season of Stranger Things while collecting these samples, because you sort of set up the mill, it does it for you scrape the sample, do the next one, get the powder. So we wanted to see what is the Venericord response rate, because their size didn't change across the boundary. But you could, similar to the extinction turnover thing, you could have no size change, but have a growth rate change. Live shorter, but grow faster, and vice versa. So here are our Venericord bivalves. So this is from the Paleocene side of things. Again, lots of seasonal cyclicity in here. These clams grow to about 130 millimeters, so about 13 centimeters. This is a pretty big clam, the size of like the palm of my hand here. And they live about 25 going on 30 years, right, these Venericords. And so you can see the first couple of years of growth, this is the first 5 years here, they're growing quite rapidly. There's a lot of space between each of these summer-winter kind of cycles, and then that growth slows down later on in life. So that's the Paleocene side of things. On the Eocene side of things, the growth is really fast in those first 5 years, but they don't live quite as long. They live to about 13 years, but they grow to a similar final size. So when you're holding a Paleocene Venericord and an Eocene Venericord, they don't look really different, but they're doing something different in terms of their life history strategy. They're growing a lot faster, but getting to that same ultimate size and living a shorter lifetime, which is interesting because that could be a suggestion of, you know, getting to this adult size really fast. You can turn over and put your genes into the next population to get to that adult size because you know that you don't have a long time to live, versus this strategy here is, oh, I can spend lots of time getting to adulthood before I need to worry about reproducing because conditions are fine. So that could be an explanation for this. But caveat is, we only did one on the Paleocene side and one on the Eocene side because these samples are 20 bucks a pop. And nobody has the money to do a gazillion clams. So as more people study these two boundaries, or the Paleocene and the Eocene, it'd be interesting to see how much this changes or doesn't change. So if you've been following along at home, when we look at turritellids versus Venericords, we see a big conflict here, right? Our turritellids, we saw extinction, especially the large species, and we saw origination at small size, and that small size was driven by slower growth on the Eocene side. We looked at Venericords, I didn't show this, but there's a slight origination of Venericords on the Eocene side, no size change, and slightly faster growth. So again, it was sort of like, there's no substantial trend here that I can pin. So I was like, we need a tiebreaker. So I was at PRI at the time, and it's this huge collection, all these Paleocene, Eocene units. Warren was teaching a paleo class. He was like, pick an undergrad, have them measure everything you can find. So that's what Isabella did, was measured all of these Paleocene and Eocene clams and snails that we could find enough of to kind of run some statistics on. So we have Athleta, Calyptophorus, which is this beautiful stromid with all these crazy like knobs that the shell grows. Natica, which is like your moon snail. Pleurotoma is another gastropod, and Chelina is a small clam. And this is a lot of data. Again, our 2 Paleocene units, and then our 2 Eocene units. So we didn't have anything from the OTB beds. And you're kind of like, do I see a change here? Let me help you. Okay, we see 2 groups, Pleurotoma and Calyptophorus, that show a significant decrease in body size across the boundary. And if you look at our N numbers here, they're not great. We've got like 30 here, 50 here, 14 up there, 50, that's 9, that's 43, right? There's a slight shift in the size distribution of these groups across the boundary. So maybe there's a little bit of size reduction in the Eocene. So one of these days, I'll get around to measuring all the clams and snails from the Paleocene and the Eocene. Another open project if you're interested in figuring out if there's actually a change in this boundary. So, okay, all taken together, we don't see this change in richness, right? No taxonomic turnover, no change in extinction rate or origination rate, no preferential selection for mollusk physiology. Nothing is really changing in a way that we can say it's directly related to the PETM and not something else about a broader trend of infaunalization through the Cenozoic. And no clear trend in these life history traits. Maybe a hint of something being different across the boundary, but nothing substantial. So we're not seeing a big selective pressure on mollusks. Or if anything, it's really weak and very specific to certain groups. Like turritelids seem to be kind of sensitive, but the venericards just kind of blow right through the boundary, no problems. So why is that? Because I did start out by telling you about these huge migrations in mammals and changes in plant communities and extinctions in corals and shifts of planktonic forams. So why aren't the mollusks, the level bottom groups, responding in the same way? Maybe they're pretty well adapted already. You know, they're already into the Cenozoic. They've gone through lots of extreme temperature events, the PT boundary, the TJ, the KPG, depending on what your interpretation is of what caused that extinction, right? So they've really been hammered by lots of events. So maybe these are the best adapted groups that are really physiologically adapted to high temperatures. They've been living through the Paleocene, which is already a really warm time in Earth's history anyhow. And so this slight increase in temperature, again, not at a tropical location, right? We're at the Gulf Coastal Plain. It's not a tropical site at this time. So maybe the temperature change wasn't great enough for them to really feel that impact. And so they were able to endure the PETM. If you look at modern mollusks, gastropods and bivalves are pretty resilient to extreme temperatures. They can live in waters 30 degrees Celsius and even higher, whereas corals are a lot more sensitive to extreme temperatures. So not all groups are as well adapted to these extreme temperature events, or not even super extreme if you're at temperate localities that become sort of warm tropical. It's not too bad. The other idea is that the shallow shelf environment may have been slightly buffered. So recent work that's come out of the Atlantic coastal plain has looked at what was the response in terms of acidification of the shallow shelf systems to the PETM. And what they're finding is not a big signal for acidification. So that big loss of carbonate that I showed you in the isotopes at the beginning come from these deep sea cores where they're seeing the rise, the shallowing of the CCD, of the carbonate compensation depth, in response to changes in carbonate chemistry in the ocean, but they're not picking up on that signal in some of these more shallow shelf systems. And so when we think about the Mississippi River or the Proto-Mississippi dumping all this sediment into that deltaic system, there's a lot of mineral deposits that can be buffering any sort of acidification that could have occurred due to this rapid shift in carbon content, CO2 being put into the atmosphere and therefore dissolved into the oceans. And so they may be buffered by these really big changes. So the temperature change may not be as great here. The acidification may not really be a problem for them in this area that's really well buffered. So those are sort of the explanations that we have for this. But again, that 1 million year hiatus really hangs over all of this. So we could always have some sort of transient response of major migration up and down the coast. Florida's not really a problem here, so they could have migrated up the coast and come back down in a million years. And we're not really picking up on that signal. So there's more work to be done. And I'd just like to leave with talking about this modern climate change has no geologic analog, right? We look back at the PETM to say, what could we learn about what we might expect in the future? And so we have this sort of best case scenario that I bring to you today, which is that you could still eat clams and escargot on the Eocene side of things. Maybe your escargot got a little smaller, by the pound, but your clams are still doing pretty good, right? But the PETM is still 10 times slower than what we're doing today. So it's a best case scenario for a not so bad scenario, right, relative to what we're experiencing right now. And on that, I'll happily take any questions and comments. I have lots of acknowledgments for all the different people. Yeah.
I really liked the presentation.
Thank you.
It was really interesting and I was able to follow along. But I just have a question. The geochemistry that you showed in the beginning with the CO2 and oxygen, were those gathered from the sediments where the fossils were found from?
Like this, I'm going to go back. So it's like the GEOS data.
Yeah.
Like these ones?
Yeah.
No, so this comes from, let's see, actually you can read my notes right here. I wonder if I have, so this is forams, so these are sites in the Antarctic, South Atlantic, and Pacific. So there's a bunch of different data that's going into here. So I think actually if I show it, it'll have the key. Yeah, so it's impossible to read because it's blurry here. But this actually says Southern Ocean, Central Pacific, and South Atlantic. So the dark blue is Southern Ocean, the black is Central Pacific, and the green is South Atlantic. So these are all ocean cores that they've taken and combined together. So they're just kind of showing that as a global phenomenon that's not just related to one particular ocean basin. And so a lot of the work that was originally done in the PETM came from these ocean drilling projects where they were just like looking to see how has the Earth changed through time, and then they pull up these cores and run the analyses. They're like, something weird happened there, and that sets off all the research into like the groups, the terrestrial fauna and the marine fauna. Does that answer your question?
Thank you.
Cool. Yeah. Yeah.
Good question. What, maybe I'm just answering my own head, but what I was going to say is, what if you ran the same kind of data analysis, but from a high latitude?
Yeah.
But the Paleocene was pretty warm, so maybe it wouldn't make that much of a difference.
Oh.
Do you think, I guess, well, do you think you would...
But you would see more warming at a high latitude,
Yeah,
even
so
though it was
you might see
warm.
more of a dramatic shift across the boundary?
There's two things. There's two answers to this question, or maybe three answers. One is I don't know of a locality, but I haven't looked for one. I haven't thought about that. Two is I sort of left the PETM behind because it's a little like competitive, and I didn't want to I felt a little bit like stepping on toes entering into it, and so I was a little like, okay, for like my first couple years as a professor, I don't need to like get into fights. I'll do something that doesn't involve like fighting. And then the other thing is I am really interested in like finding some boundary sections, and I talked to some people at UCSC, like Jim Zakos, about like, tectonic basins. Like there's got to be a couple basins someplace that are keeping up with these like, there's a global regression, so that's why you don't see a lot of the actual boundary sections, but I don't think that there's no boundary section anywhere on this planet that records the shallow marine setting, so there's that too. But yeah, it'd be very interesting to see, because even if we could do a similar study, even with a hiatus at the boundary itself, just to see what the after effects are, if there's lingering change, and I'd love to do the same thing at the Atlantic coastal plain, which hasn't been done either, to see what's changed there. Because that's how you could really track a migration, is just to look at different sections of that coast.
When were there a lot of regressions, like during the boundary?
Yeah, so the reason that there's no known boundary
Right.
section right now is that there's like a regression during that time. It's like a relatively low sea level.
Why?
Just like, because like all the different things that go into sea level rise and fall. So nothing necessarily associated with the event itself, supposedly, right? But if you know about common cause hypothesis, there's this idea of like what changes the biology is also changing the geology.
Right.
So this like, if there's a regression, maybe that causes the clathrate disruption that causes the PETM, right? Like, so there could be an association with those things, but the regression is maybe like geological, whereas this is driven potentially by a geological phenomenon that's causing this extreme, change in carbon cycling.
Yeah.
But potentially unrelated.
Interesting.
Has anybody ever looked at any of the terrestrial snails to see if there's any sort of correlation between how those are responding with, instead of just the shallow marine?
You know, the answer is yes. And I think I have that paper, and I think I've never read it. Yeah, because I don't think it was like a big change either. Otherwise, I would have like written about it. Yeah, that's interesting. Let's look in my file. I'll take this off and look at it. Yeah. Somebody gave me also like a box of like Paleocene or Eocene snails, but they didn't have like collection locality information. I was like,
Tasty.
Yeah. Yeah. Teaching specimens.
Right.
That becomes, yeah. Other questions, thoughts? I always love questions because I get great ideas for like things I should do in like 3 more years.
I'm stuck on one thing, just a comment.
Yeah.
Is thought Svalbard had
A PE
pretty
section?
rich Paleocene section.
Yeah.
I don't know if it-
Where is that?
Svalbard.
Oh, in the
Yeah,
Arctic.
Oh, we're in Svalbard.
I don't know where Svalbard is.
Oh, sorry.
Yeah.
Svalbard is currently a very, very high latitude island in the North Atlantic, I guess North Sea. I don't know who owns it anymore.
Russia? Or is it its own thing?
Sweden, maybe. Anyways, but it has a really rich marine sedimentary section exposed. But it's
That's
one of
where
the few places I know that has a good Paleocene record.
Oh, interesting. Because it's a PT boundary there. I've never been. It's also where the seed bank is, right? Or the world seed bank is in Svalbard, I'm pretty sure. Yeah.
There's one in Colorado.
Yeah. Yeah, lots of polar bears. I really want to go. There was a PT like big like not a meet. There's like this big like 10-year meeting that like met every year, and they had a field trip every year. There was a big field trip to Svalbard while I was a grad student, but I did not have $6K to drop to go to Svalbard, so I missed that one. Yeah, that's an interesting. Yeah, I keep a folder of like future PETM. Like there's places in Nigeria that look like suspicious to me. Yeah. I'm just always looking for like late Paleocene, early Eocene units where maybe no one's looked for a boundary.
Hey Todd, do you know does the Moreno Lodo capture the big Eocene boundary?
I don't know. There's the Cranehagen is all over the place there. That's marine.
I'm so sorry to do this, but I need to cut it off because I know students need to run on
Oh.
to other classes.
Yeah, no worries.
Sounds like you might have 5 minutes or so
Oh, yeah, I
to
got Nordic.
chat. So if you want to hang out and chat, but let's thank
Yeah.
Carlie one more time.
Thanks all.